Ladies and gentlemen, we'll go ahead and get started again in just a moment. And thank you very much to all of the presenters thus far. It has been a fascinating series of presentations, and to Kim Sampson for chairing the last session. I am very proud to announce our chair for the next session, Professor John Tam, longtime stalwart of APACI. And just a reminder, Professor Tam will also be involved in the WIC 2026 conference coming up this weekend in Chengdu. If anyone is interested in joining and has not registered yet, there is still time to do so. Thank you very much. And John?
Thank you, John. So welcome back. And we will now begin session 2, which is on RSV, COVID, pneumococcal, and emerging threats. I'm John Tam, and it's my pleasure to chair this session. So just a reminder, make sure that when you ask questions, use one of the mobile microphones so that people online can understand and access to your comments. Of course, like the first session, we will have a warning for 3 minutes and 1 minute remaining to help us track time. And now we would please join me in welcoming Professor Dhanasekaran, faculty member of the School of Public Health at Hong Kong U. Professor Vijay, yes? Please go ahead.
Good morning, and thank you so much to the APACI organizers for repeatedly inviting me to give a talk at this meeting. And I was present at the last in-person meeting with Singapore, which was wonderful as well. And considering this was on respiratory diseases, I somehow managed to add all 3 major respiratory virus names on the title: COVID, flu, and RSV. So today I'm going to talk more about influenza and RSV and the work we've been doing at the Hong Kong School of Public Health. A lot of this work is done through global data sets developed by global genome sequencing and several epidemiological studies. The RSV work I'm going to present is basically focused on Australia, Australian data, but also WHO has advanced RSV surveillance globally in many countries, and so I'll be also using that data a bit. Okay. Oh no, something wrong.
Sorry, Vijay.
It's all right.
Technical glitch. No worries, John. Great, thank you.
So continuing on, immediately after COVID, as we all remember, Hong Kong and mainland China mostly had a zero COVID policy, where it was pretty much contained through the first 2 years. We've had sprouts of outbreaks in the first 3 to 4 waves, which didn't cause enormous numbers of cases. So I had some time to look at influenza at the same time, because influenza surveillance was continuing during COVID. A lot of resources from influenza was transferred to COVID during the pandemic. However, the testing actually increased for flu as well at the same time, and there was enormous testing for COVID, so it was very easy to implement simultaneous testing for influenza. And we used that data set to really understand what was going on, because influenza, as we know, is seasonal. In the northern hemisphere, there's a stark season as you go in the north. Similarly, there's a completely different season in the south. And in the tropics, the seasonality is a bit more complex, with introductions from both the regions from north and south, as well as local factors and density, population density, the migration, and lots of other factors contributing towards the seasonal patterns that we observe in the tropics and the subtropical regions. So this plot on the left shows the changes in the genetic diversity of each of the 4 influenza lineages immediately after the pandemic. So before the COVID pandemic, around 2019, H3N2 viruses and H1N1 viruses had diversified enormously. And I have to highlight that the H3N2 viruses in particular is renowned to evolve antigenically very quickly, in the sense that every 3 to 5 years, we have a new antigenic variant emerge. And hence, what that does is it eliminates all other variants, hence only one single lineage persists. And that is the main driver for updating our vaccines every year. However, since the 2009 swine flu pandemic in 2009, the H3N2 viruses had started to diversify, in the sense that we haven't had a very dominant H3N2 virus lineage for many years. Similar for H1N1, since it's emerged in 2009, the clade 6 became the dominant clade, but then since then, it had started to diversify and there were many subclades. And we were having some uncertainty in terms of selecting the right vaccine candidate to be incorporated into the vaccines, because 30% would be one of the subclades, although their antigenicity was not too different. So people were happy to pick viruses based on the best virus, which grows into a vaccine, grows into a cell culture, grows into a vaccine. However, since the implementation of global control for COVID in April 2020, the genetic diversity largely collapsed. One of the B lineages, sublineages, B yamagata, was never detected since that time. B Victoria continued to circulate, and H3N2 and H1N1 had diminished in diversity. So we wanted to use the genetic data available at that time, as well as epidemiological information about location, data of isolation, and things like that, to understand what really happened to flu during COVID. And ultimately, we found that there were some pockets. There were some pockets where some lineages had continued to circulate. For example, if you focus on B Victoria, there's one yellow-colored tip dot on the tree there, which is predominantly only found in mainland China, because mainland China was zero COVID, but not zero flu during the time. Nobody was getting out, getting in, and it had a large enough population size to sustain a single lineage of B Victoria viruses over 2 or 3 years before it could again escape the country after COVID was completed, and it started migrating globally. So at the end of COVID, we essentially, before COVID on the left, we sort of had a mixture of genetic diversity globally. As you can see, multiple colors coalescing. But also there's some lineages like Australia, we mostly see one particular clade. However, after COVID, we see that each lineage is sort of managed to sustain geographically distributed. For example, Victoria viruses, again, there's a unique diversity of viruses in mainland China, Africa, and we find that some regions such as Southeast Asia, West Africa, some parts of Europe were actually sustaining populations despite the control measures. And some of these lineage viruses, these are individual bars that represent the number of cases that we detected among each of the lineages that circulated. Most were singletons, but some did infect, and we got hundreds of sequences, and we could find out which sequences sustained and evolved during the COVID period. This is the current diversity of H3N2 viruses, for example. As you can see from the phylogeny, we'd had viruses that were quite diverse, circulating viruses that has diverged before 2015, for example, had been detected until 2021. And the diversity is kind of like sparse. It's not really constrained, and there's not like a clear antigenic jump. except the orange branch called K, which is the most newest antigenic variant, and that antigenic drift event seems to have occurred after many, many years in H3N2, and that's the current situation of H3N2 viruses. So just giving you an idea that how migration dynamics, persistence can affect decisions in terms of vaccine selection and vaccine administration. So moving on to RSV, I'm not going to talk about the burden of RSV and the control mechanisms towards RSV because I just noticed the next speaker is going to focus on that one. So I'm going to move out saying that immediately after, during COVID, Australia had a huge off-season RSV outbreak. And RSV is usually starkly seasonal in Australia. It occurs mainly during the winter months. And as we've noticed, we started observing outbreaks in multiple states. New South Wales, Victoria, Western Australia, and we also found different lineages of viruses causing the major outbreak in Western Australia and New South Wales. What was most surprising to us was that the lineages that we've detected in New South Wales, ACT, and Victoria, these viruses had several amino acid mutations which we had not detected and observed in the global surveillance for many years at that point of time, as illustrated by all those amino acid marks on the right tree there. So as we can, and we used molecular clock methodologies to understand what is the time of the most recent common ancestor, which sort of usually gives us an indication of the time of virus introduction into the region. But our analyses actually showed that these viruses have been circulating in the country for a while, but had not been detected, which sort of gave us a suggestion that these viruses must be locally persisting rather than like flu, where every flu epidemic literally happens by viruses repeatedly being introduced into the region, and small epidemics leading to larger epidemics under conjugal conditions of good weather and lots of contact patterns, and the R naught is more than one, for example. Whereas in RSV, they seemed like patterns were slightly different, there was more persistence, and it wasn't clear, but we did publish this immediately to highlight that RSV could cause large outbreaks during the COVID pandemic. New Zealand published a study a couple of years later with a lot more data accumulated over the years. As you can see, the regular graph shows strong seasonality in each of the years before the COVID pandemic. But then immediately after that, New Zealand, the quarantine measures were opened down, and then New Zealand had a large outbreak. And from phylogenetics, we were able to find out that, phylogenetics, they were able to find out that the viruses which had circulated in Australia had been introduced into New Zealand and caused the outbreaks. So introduction of these viruses in New Zealand caused these outbreaks, whereas the evidence shows that there's not been any endemic lineages in New Zealand for the first time, whereas in Australia, we had observed endemicity of several RSV lineages. So taking all this data together, we again collaborated with Ian Barr, mainly heading from the WHO CC in Melbourne, with a bunch of other laboratories from other states. We sequenced a lot of viruses from that period from 2019 to 2024, about 4,000 viruses, and we also contextualized that with 24,400 global sequences, taking advantage of the WHO initiated surveillance. So since 2016, WHO incorporated a pilot RSV surveillance combined with the GSIRS, the Global Influenza Surveillance and Response System, in 15 countries. And after a pilot, it's been expanded to much larger countries, and especially the developed countries are sequencing a lot more viruses. But I also want to highlight that there are some regions which are still very undersampled. For example, South Asia, India has very few sequences compared to the population size. China has just started sequencing a lot more sequences, but there's still very less. So there's still large pockets of regions without much surveillance. So I'll explain the caveats as we go. And again, I just wanted to highlight that the outbreaks that's happened in Australia in different states since the COVID-19 pandemic have been asynchronous in the sense that usually the peaks match quite nicely between the states, especially in the southern states, except the Northern Territory, Darwin, which exhibits more like a tropical signal. Most of these states are synchronized, but here you can see that since 2001, there are some states which match, but there's not really a clear synchronicity among these outbreaks. So what this really means is that just by having one or two sentinel sites across the country, you can't really predict or determine if there's going to be an RSV epidemic which is commencing. So that's the main problem with RSV, that in flu, for example, we can pick 5 or 6 cities around each global sites, and we can start doing intensive surveillance, and we get a good idea in terms of what sort of virus is circulating. Whereas for RSV, it looks like we may have to find more sentinel sites to be able to give a better understanding of what diversity is going on. So these are the hypotheses that we had at the time, and we started analyzing more data. And as you can see, these outbreaks in Australia are actually all derived from big clusters, long branches leading to it, which indicates that there's lack of genetic diversity that we've captured. The outbreaks are mostly localized. So you see the orange blobs from Western Australia localized. The blue ones from New Zealand are localized. The green ones derived from Western Australia to Victoria. They kind of some regional movement, but a lot of these outbreaks have been localized in multiple outbreaks, as we continually see. So we try to better understand what are the factors of these viruses in terms of persistence and quantifying the persistence. And as you can see in the graph in the middle, that we do find lineages persisting about 2 years or 3 years in some states. Some lineages are persisting for more than 2 and 3 years, which we've never really observed in influenza viruses. So taking all this data, we looked at both RSV B and A. Immediately after the COVID-19 pandemic, RSV B viruses are becoming more dominant. So we focused on the major 3 lineages, A, and B lineages, to better understand these dynamics among the globally circulating populations. So this is the data that's available globally from different continents and it's colored by lineages. And asynchronous outbreaks also suggests that there are multiple different lineages that are actually co-circulating at the same time globally, and they are actually persisting in these individual continents rather than actually mixing globally all the time. So focusing on the bottom graph here, you can sort of understand that Australia, for example, for BD41X, Australia was a major source of these viruses, maintaining these viruses for a period of time and then exporting to other continents. Similarly, for the second one, 83X, Asia acted as a major source of these populations. So each of these lineages, we could find some places that were dominating. But the key thing was the genetic diversity that was observed before the COVID pandemic is continued to circulate after the COVID-19 pandemic. It's just that no lineages has gone extinct for RSV since that time period. This is the same graph as the previous one, where you can see that BD41X is predominantly seen in Australasia region, 81X and 83X mainly in Asia and Europe, and things like that. So ultimately, taking all this together, we found that there's regional endemicity, which actually shapes the global landscape of RSV. It's pretty clear. The duration of circulation is actually decoupled from the transmission intensity from a region. For example, if there's no viruses circulating, no viruses coming into Australia, for example, there are some lineages within Australia which is undetected, can emerge to cause an outbreak at any time, considering accumulation of neonatal cases or pediatric cases without any antigenic evolution that is needed. So it's just possibly a mechanisms of lowering immunity, sustained circulation, potentially in minute spots, maybe in a cluster of schools, maybe in an eco-center, where we really need more data to understand where these lineages might be localized. So ultimately, this is a graph showing that the global diversity of RSV in 2020 did not diminish at all. It sort of has become more diverse than before. And so ultimately, all this leads us to indicates us that there's more complex ecological drivers maintaining RSV circulation in Australia, for example, in our study, but this could be the case in many of the cases. The evolution rate, this has been well established that the F protein evolves much faster, but we found that there's no evidence of selection pressure on these viruses to escape anything, so which also indicates that things which are sustaining are actually continuing to circulate in subsequent years during COVID without any effect. So taking all this together, seasonal reintroductions may not be strictly required for RSV maintenance in temperate regions. Unlike flu, RSV clades can persist regionally for more than 10 years. This is at the continental scale. This long-term persistence may explain the limited generic bottleneck after the pandemic. So geographically, so why this matters for public health? As I mentioned earlier, geographically granular genomic surveillance is essential for tracking resurgence dynamics, and better regional surveillance can support vaccine and prophylactic development strategies. Thank you.
Thank you, Vijay.
Thanks, sir.
Interesting. We have any questions from the floor here? No? Or maybe we have a question online, and no, there's no question online. You're such a good presenter. Everybody understands.
Thanks, Prof. Tam. Makes me feel good.
Okay. So thank you very much. So our next presentation is going to be online. It's from Professor Tantawichien from Thailand. It's the Division of Infectious Diseases, Faculty of Medicine at Chulalongkorn University. And his presentation is on advances in RSV and pertussis prevention in adults. Please go ahead.
Thank you, Mr. Chairman. I'd like to thank Asian Pacific Alliance for control of Influenza that invite me to talk about the ISV and pertussis prevention. May I share my slide? So today I will talk about the advance in ISV and pertussis prevention in adults. This is my disclosure. I work for the many research on the vaccine, and also I work on the new pertussis vaccine, and also work for the ISV manufactured by Janssen, but it's not to the marketing. I think today, when we talk about the advance in ISV and pertussis prevention in adults, I have the three topics today. The first one is the burden of ISV and pertussis infection in adults and elderly. And second one is the progress in vaccination for RSV and recommendation. And finally, I will talk about the pertussis vaccination for adult and elderly. So when we talk about when we talk about the vaccine preventable disease for elderly and adult, we have to think about the vaccine that prevent LRTI or pneumonia, because the pneumonia is the leading cause of infection death in the elderly group and people who have the medical comorbidity. So vaccine for LRTI and pneumonia prevention. Influenza, we know, COVID-19, pneumococcal, and today I will talk about the RSV and pertussis vaccine in the form of the Tdap and pertussis. So when we think about the viral infection in RTI, we call the tripledemic virus, influenza, COVID-19, and RSV. You can see that the flu, COVID-19, and RSV is caused the illness in elderly, especially increase of the infection and hospitalization and death. Most common in flu, but COVID and RSV are also common in the elderly group, and it causes hospitalization and death in this group. I will start with the RSV, burden of the RSV. RSV is a virus that can infect in our long life, okay? Infants, children, adult, middle adult, and also elderly. And when you see the infection is most common in children, and it's called children need to hospitalization because they have the wheezing, they have the hypoxemia, and need the nebulizer. But in children, older children, and adult, is no problem. It's just only the URI. But in elderly, someone elderly may have the severe illness, such as the exacerbated COPD, exacerbated asthma, heart failure, and also pneumonia, and it caused the hospitalization in this group of the patient. And you see the data in Thailand show that it is some of the elderly need to hospitalization because of the RSV infection. When you compare between hospitalization and mortality, in infants it's high rate of the hospitalization, but in elderly it's high rate of the death in this group. So it's either one of the viral infection that important in elderly group. The Spain had a study about the risk of the hospitalization that's caused by RSV. The first one, you can see that elderly group, especially over 75, and the other comorbidity that is cause the complication in this group and need hospitalization. The first one is the asthma, COPD, cardiovascular disease, and also hematologic cancer. But the other comorbidity can increase the risk of the hospitalization caused by RSV. The Thailand had the study about the adult who have the viral infection that need hospitalization. They found that 5% of adult hospitalization suspected viral RTI is caused by RSV. And most of them is an elderly group over 50. And you can see that when RSV infection in elderly group and need to hospitalization, okay, very interesting, 50% have the hypoxemia and 1/3 need ventilation. It means they have the respiratory failure. And most of them, 80%, they have the pneumonia. And some of the patients have the complication, cardiovascular complication. And you can see that the mortality rate in this report is around 50%. Recently, we also have the RSV hospitalization reported from Thailand, nationwide retrospective observation study. We found that case fatality rate of the RSV hospitalization is around 9.5%. And mortality independent as with the age group more than 75, length of stay over a week. The patient who need ventilator and also who have the pneumonia and acute MI. And you can see that survival, 60-day survival of this group is decreasing rapidly in very elderly group and also the patient who have the comorbidity. So it means that it is the same as the European country. Thailand and many countries in Asia also have the problem with the RSV in elderly group. The other disease that we talk today about the burden of the pertussis. We know pertussis is a highly contagious disease. Okay, R0 is around 12 to 17. It's the same as the varicella. Okay, so the spreading of disease is very easy. However, when we talk about the pertussis, we're afraid of this disease in infants because it can cause the apnea, hypoxemia, and respiratory failure like this. Okay. However, when this disease occurs in adult or elderly, it may cause the prolonged cough in this group of patients. And many patients did not receive the diagnosis by this disease because they need the PCR and complicated case. It may be difficult to test PCR for diagnosis of this. So it's called the delayed clinical diagnosis. So when we see about in the world, increasing of pertussis case occur, although we use the pertussis vaccine for infants for a long time. Example in USA and in China, okay, and after COVID, okay, after COVID, many in Asian countries reported increasing of the pertussis case in Asia country. They show that increasing of the pertussis case after COVID in Japan, in USA, in Australia, and New Zealand. Okay. However, most of the cases occur in children, in infants, and also in adolescents. However, you can see that some cases still occur in elderly group or adult group. That is mean we have the spreading of the disease after COVID-19 era. In European country or in many countries, they found that the chipping of pertussis group occur in adult, around 10 to 30%. of the pertussis cases occur in the down. Okay, you can see this one. Okay, shaping of the around 10 to 30% is occur in the down. But I can say that adult with cough is not easier to make pertussis because they have to care about the definition. I mean, cough more than 2 or 3 weeks, and need to send a PCR to get the diagnosis. So we can think that prolonged coughing is underdiagnosed pertussis in our practice. Okay, Thailand, we also have the study about the 76 Thai person who cough more than 2 weeks, and then found that 1.7% PCR positive, and probable case is around 18%. So it means in our practice, it's very high case of pertussis that we don't know. We think that it's maybe bronchitis, oral bronchitis, maybe mycoplasma, anything that patient will cough a lot. So pertussis in the down is an underestimated case. You could like the tip of the iceberg. I think when you practice in the clinic, you may see the patient. You can see in another patient in our practice. Okay, but sometimes we don't think that they have the pertussis. Okay, so the reported of the severe pertussis infection in US and in many countries. Usually severe pertussis it occurs in infants because infants who have the pertussis, they may have the apnea, they may have the hypoxemia, but increasing of the severe pertussis occur in elderly group like this. You can see that it's increasing of the severe pertussis in this one, and some they need hospitalization. Okay, so among older adult hospitalization for pertussis, 90% they have underlying medical condition, especially lung disease, asthma, COPD, and some may have the obesity, diabetes, cardiac disease, and also smoking. So this is the risk of the severe pertussis. If they have it, if the patient have this underlying disease, it may have the severe pertussis. Okay. In the study, pertussis in high-risk group, example, asthma, COPD, immunocompromised host, and also diabetes and obesity. So we can imagine that if pertussis, okay, infection in this group of the patient, I mean, COPD, asthma, diabetes, cardiovascular disease, immunocompromised host, it may cause a severe illness and may need hospitalization, and may have the complication such as the pneumonia, aspiration, lip fracture, and maybe carry serious illness. Next, second topic, I will talk about the vaccination for RSV and recommendation. If you know about the vaccine, RSV vaccine, they need nearly 50 years to develop this RSV vaccine. It's not easy to make this vaccine in our practice, okay? Since 1957 until we have it now, okay? So it's not easier to make this vaccine. Now, we know that PF14 is really good for make the vaccine for RSV protection. And in the world now, they have the 3 band. They have the 3 types of the RSV vaccine. The first one is the recombinant with adjuvant by GSK. Second one is the recombinant IVPF antigen by Pfizer. And the other one is the mRNA vaccine from the Moderna. This vaccine that tests in phase 3 efficacy study, and it showed that vaccine efficacy in every is look high. It's allowed 70 to 80% to protect the people from lower risk infection, and may higher, I mean nearly 90% to protect them from severe LRTI. That's important to us that if you use the vaccine, you can protect someone may have the severe illness, RSV elderly. With the in Thailand, we have only the 2 types of vaccine. I mean the adjuvant RSV and non-adjuvant RSV. Okay, we found that in the efficacy study for 2 or 3 seasons. Season is mean they test in the winter season because in the European or US country they have the RSV in the winter season. But in Asia we have it in rainy season. Okay, So they found that the efficacy to protect them from LRTI or severe RTI is over 80% and 90%. And protective effect is really high in serious illness, such as the severe RSV LRTI. And also the patient who have the medical comorbidity, it can protect them very high, like the 80 to 80%. So in my opinion, this is look very good for the vaccine to protect the RTI in our practice. Okay, so recommendation I like to show in the USA. US recommend to use this vaccine in all adult age over 75. That you can see that like the same the study in Thailand and the study in Spain. Okay, over 75 is higher risk of the severe complication and death. Okay, I think age over 75 is the appropriate to use this vaccine. And US also use in 50 to 74 who have the high risk of complication, such as this underlying disease, cardiovascular disease, cardiopulmonary disease, and maybe immunosuppression and other medical comorbidity, the liver failure, corneal failure, diabetes, okay, may need it, okay, and you can use it in any time of year, okay. So this is a recommendation in USA. However, many countries may have the recommendation are different, okay, such as the age-based. Many countries think that over 75 is very appropriate to use it, but some countries use in over 60 or 65, okay, and some countries also use the risk-based recommendation, such as in US use in the risk-based is over 50, but most of the other countries use in over 60 who have the medical comorbidity or immunosuppression. They need to get this vaccine. However, this vaccine is was used in around 2 or 3 years in USA. They also report about the effectiveness of the vaccine to prevent RSV hospitalization. Around 2 seasons, first year is protect around 80%, okay, but the second year is protect around 60%. That is so good when you compare with the other vaccine to prevent RTI, such as the flu or pneumo. This is a higher effectiveness when compared with the other vaccine. So I think this vaccine is very good to protect RSV. The last one is the pertussis vaccine for adult and elderly. Okay, you can see that we like to protect pertussis infection RSV in infants, because if infants have the pertussis, they may have the serious illness. I like to show you about the apnea, hypoxemia, and maybe death. So now we use the pertussis vaccine for infant the first year of life. In Thailand, you use the DTP on 2, 4, and 6 months. However, in the early of life, they may need the protection at the time of delivery. So the pertussis vaccine for adults for use in maternal vaccination to produce the antibody and pass the placenta to the infants in early of their life. So global pertussis vaccination, they will concern the first one that we have to increase the pertussis protection in infant and toddler immunization that we use in all. And you can see that in Asia, DTP 3 doses cover in Asia is really high, except some country, over 80 or 85%. Okay, some country is over nearly 1%. Okay. Second thing that we need to use the vaccine in pregnant women between 20 to 32 week gestation, because we like to deliver the antibody to the infant in early of their life, because it can protect them in the earlier, maybe within 3 months after delivery. Okay, that's really important to infant to protect them from pertussis. Okay, and the third, we encourage the healthcare personnel who have direct patient contact, RSV neonatal care. Okay, because neonatal may get the pertussis from the healthcare, so healthcare who work in neonatal ward, okay, they need to get the vaccine because they need to protect pertussis infection, because they can spread to the neonatal or infants. Okay, the last one maybe pertussis Tdap or pertussis to all other adults. That is very expensive. Usually we use the tetanus diphtheria in all adults, but someone think about that we may need to have the pertussis prevention in adult. Okay, Tdap or pertussis in pregnant women is very high efficacy. When you can use this vaccine, they can reduce the pertussis in infant very rapidly, and efficacy of fitness to protect them is in infants is around nearly 80%. So pertussis vaccine in pregnant is very good. It's high effectiveness that many countries try to use it in pregnant women. However, we may need the boost of the pertussis for life, especially adolescent adult. I showed you that older and the adult who have some medical condition may have the complication from pertussis. So we may need the boost pertussis immunity in this group, I mean adolescent and adult. So GPI recommend for low middle income country, many countries in Asia, they try to do that. We have to increase the coverage of the pertussis in infants, the first thing. Second thing, you may need immunization in pregnant, okay, if resources allow, because if you have the money, you need to put the pertussis to pregnant. That in Thailand now we use it free. Pertussis vaccine is free for pregnant to protect the pertussis in infant. However, when you see the WHO think the same thing, you have to need the pertussis protection in infants. US, they use all adult. Or they think that the adult may need the pertussis at least one dose to protect them from pertussis. And you can see that the China Global Initiative for Asthma, they recommend the pertussis for adult and elderly who have the asthma, because they know that asthma, if they have the pertussis, they may have the severe complication. Okay, so pertussis vaccine for adult now, many countries use adult or boosting. Okay, I mean that they use for adult for a long time and elderly for a long time. Okay, you can see that Australia, they recommend the Tdap, I mean pertussis vaccine with diphtheria and tetanus in adult, and in any adult who need tetanus contained vaccine, they use Tdap, replace tetanus or TD. And in Taiwan, they use the TD every 10 years. Okay. However, most of the country, they still recommend to pregnant women. They success in the infant protection, but now they move to pregnant women need to get it. Okay. However, in European country and many countries in Europe, okay, they still recommend the Tdap every 10 years. It means that they think that the pertussis is maybe important in adult and elderly group. Okay, so now we have the new pertussis vaccine. The other platform of the vaccine we call that the pertussis toxin, genetically detoxified pertussis vaccine. Okay, that produced by Asian country. Okay, and we prove that we not public now, but we prove that this recombinant pertussis is look better. It's produce higher immunity and longer immunity when compared with the other cell or Tdap. That is chemically detoxification. This is genetically detoxification pertussis toxin. And when we use it in 18 to 35, the recombinant pertussis vaccine is look good when compared with the old vaccine. Okay, so we can see that Malaysia, Singapore, Thailand, okay, we start to use the pertussis vaccine in pregnant women. Okay, and Thailand now we recommend every 10 months. You can use TD or Tdap every 10 months in Thailand. So finally, I think that RSV and pertussis infection is common, or we have to prevent this infection in some hosts, elderly and adult with medical comorbidity. And now RSV recommendation usually used in over 75 and the people who have some medical condition or medical comorbidity. And pertussis vaccine can use it maybe every 10 years, but it costs in our population. But some people, maybe elderly to people who have the asthma or severe immunocompromised host may need it. Thank you very much.
Since this is an online presentation, we will have no questions and answer session. And we'll carry on. Next talk is going to be from Professor Tommy Lam. Unfortunately, Professor Tommy Lam is unable to join us today in person. So he will be presenting in online also. Okay, thank you.
Hi, can you hear me? Hello, can you hear me?
Yes, no problem.
Thanks, Professor Tam, and thanks for a kind introduction. And I really apologize that I cannot come in person. So let me first load the presentation file. All right, I guess, can you see my screen?
We cannot see any slides.
We cannot. Press shares. Let me share again.
You should be able to share your screen.
Yeah, I already press share, and in my my screen is already is shared.
It's not on our end, unfortunately.
Okay. How about now?
Yes, it's fine now.
Okay, great. Yeah, so let me see if I can. No. Okay, cool. Great. So you can see, right?
Yes.
Okay, great. So thanks for organizer for really grateful for your invitations for me to give a talk presentations on the vector-borne disease as an emerging threat in the region in Asia Pacific, and for those who my friends and knowing my kind of research track record is that I actually start my research career working on respiratory pathogens, influenza, coronaviruses. And I then, in Hong Kong, one of the, very important area, with connections with the regions, and a lot of vector-borne disease, is ongoing in the region. So I start to research in this this group of vector-borne disease recently, in the last 15 years. And so I'm going to present, some brief introductions, especially some findings or kind of observation, epidemiological observation of this type of vector-borne disease emergence in Asia, and also some of my research finding as well. Now, vector-borne disease, is regarded as a disease transmitted by living blood-sucking arthropods, mostly mosquito, ticks, fleas, lice, etc. Rodent is considered as one of also some people also consider as disease vector. Although majority people when talk about vector-borne disease, they are talking about arthropods, organisms for disease transmissions, and this type of vector organisms play various level of role in their disease transmissions. Some are obligatory to many vector-borne diseases, and so it means that the transmissions has to go through this vector for many of these vector-borne disease. For example, dengue, and that means this type of vector-borne disease have very interesting characteristic that the disease emergent is a result of a complex interactions between the vector host, including human and animals, and also environment climate as well. Because the vector, the arthropod, is a very highly kind of depending on the environments to survive. And so understanding, these interactions will help us to inform better disease prevention and control of vector-borne diseases. And you can see on the right-hand side is one of the recent article that we reviews, one of the tick disease vector, and you can see the ticks is having a kind of complex life cycle, and then with exposure and infestations on animals, but also potentially transmit to human by the blood-sucking activities. Now, vector-borne disease, is actually a very important disease. Although I'm sure a lot of you have a lot of attentions on the respiratory pathogens diseases, and the vector-borne disease indeed contributes to about 17% of all infectious disease globally. And another very interesting characteristic that it is the pathogens include not only just viruses but also bacteria and also eukaryotic pathogens as well. So these vector can transmit both viruses and bacteria and also eukaryotic like parasite worm parasites. And some of you, immediately have in your mind is that dengue virus is one of the important vector-borne disease. But in fact, for these vector-borne disease, vector-borne viruses, there are more than 500 virus species that has been reported, has been known. For bacteria species, it's known to be less, but then a lot are still to be identified. And with the advance of the diagnostic detections and also sequencing approach, more and more these sub pathogens are being discovered. And on the right-hand side, it shows the basically distributions of different vector-borne disease, the viral and also bacteria, and also in ticks and a mosquito here. So as I said, the immediate example of dengue vector-borne virus in your mind is dengue virus, which is a feifi virus transmitted primarily via Aedes aegypti and Aedes albopictus. It's an old virus that discovered quite some years ago in 1940s, and then but it continues to explore for new territories, and now more than 100 countries has been affected, and although with it has a generally low mortality, but then untreated severe dengue could happens, and then it actually cause quite significant health burdens. And you can see that on the right-hand side is the report of the dengue virus in China on the right top corner. And so basically you can see in 2024 compared to the last few years during the COVID, it has jumped up a lot compared to the 2019 2020s. And so it obviously this is related to the control measure and the human movement in the regions. And if you look in the Sri Lanka dengue chance, it also have the similar situation that when during the COVID period, the dengue cases drop a lot. Of course, some unavailable of clinical facilities or during this COVID period may also explain such observation. But in general, because dengue is mainly transmitted through human to human via the mosquito, so human host is a very important driver for these infections. And another important mosquito-borne viruses is chikungunya virus. that recently, especially in the Asia regions, particularly in Hong Kong, in fact, because last year, some of you may know that there is a huge outbreak in China, and then also have quite some imported case of chikungunya in Hong Kong. And so it's primarily transmitted by Aedes aegypti, but some virus mutation actually help the virus to also adapt to Aedes albopictus, making these virus able to transmit and expand their territories through infecting, transmitting raw new hosts, new vectors. And this may also explain why the virus they come to China in Guangdong province particularly. It actually caused quite significant issues and also endemic transmissions there. Because we know that in China, most of the regions, the Aedes aegypti activity has been known. So the use of the Aedes albopictus as a transmission vector, could lead to the emergence of this outbreak. And we have an opportunity to collaborate with Professor Nilekar in Sri Lanka and looking into the outbreak in Sri Lanka last year in 2025. And there are a couple of previous outbreak in Sri Lanka, and through the genomic sequencing and also genomic analysis here, we can see that the 2025 outbreak, they are genetically not linked to the previously outbreak in Sri Lanka, but rather is introductions of the virus from India and other regions to Sri Lanka. And also by looking at the file geography, basically the trajectory of the virus's movement by looking into the virus genomes, we can see there are quite intense virus movement in the regions. Now, many of the mosquito virus that, as I just described, they are basically they are kind of old virus, and then there are already hundreds of mosquito-borne virus species that have been identified, and but still, the researchers are keep finding new viruses, especially using the high-throughput metagenomic sequencing, which can unbiasedly and without a prior knowledge of detecting known certain virus species, but comprehensively screen for all different type of viruses in the samples. And people still find new mosquito viruses. However, most of them are insect-specific and they don't cause human diseases. And in Hong Kong, my team has been conducting the longitudinal survey of mosquito, sampling over a few 10,000 of mosquito over the time, and then we have been using the metagenomic sequencing as well as the conventional molecular detection methods for the arboviruses. And we so far we find not many of them they are insect-specific. Only a few are actually pathogenic, and mainly they are causing animal diseases. For example, the Gitter virus and the Arcaben virus were identified from the Armigerus mosquito. And so this is some sequencing of the Gitter virus that we find in mosquito in Hong Kong, and they were found by using the molecular phylogenetics approach, we can see they are introduced to Hong Kong from mainland China and since 2018. And as I mentioned, most of the the mosquito viruses, they are quite kind of old, and we don't see a lot of new mosquito viruses emerge in the last kind of very novel one emerge. But of course, the old one, they re-emerge from time to time for the activities. For tick-borne virus, however, it's very interesting that they actually we find completely new tick-borne viruses over the time. And for example, since last 15 years, people has been finding a lot of different tick-borne viruses, and then some of them actually cause a quite significant outbreak. And the most significant one, I would from my perspective, is the severe, the one that caused the severe fever with the thrombocytopenia syndrome in patients. And it was first reported in 2009 and 10 in China with a pretty high mortality rate, mainly transmitted by the Asian long-con ticks. And initially, it was only reported in China, but now it has already been transmitted to Korea and also Japan. And here is a kind of epidemiology that you can see that the SFTSV, the report over the time in Japan, which is, you can see it's over the time is increasing. And so it's actually alerting tick-borne pathogens. Another tick-borne viruses that is very interesting, I would regard as, is the Jingmen tick viruses. That is, is widely distributed in the tick population across China. And then, in the beginning, we find it in the ticks in China. Then we, through a retrospective studies in human infections in hospital, we find that actually the virus was reported since 2000, has already been infecting patients since 2010. And then the interesting thing is probably not only just the human infections potential and risk of this virus, but also the evolutionary biology of this virus, because this virus is a segmented virus with 4 very novel genetic segments, and only two of the segments with the partially region of these have a evolutionary linkage to the flaviviruses. So basically, flavivirus is a virus with linear genomes, and these explain, this virus provide a linkage relating the linear genomes virus and then segmented genomes virus. And more interestingly, that through some collaborations in Cameroon, we find the JMTV in the ticks in Africa as well. And obviously, this is and not a very new virus in the animal population. It is an old virus, but then only recently been reported. And we have opportunity to work with mainland Chinese scientists looking into the tick virium from 31 tick species. And obviously, you can see the samples collect from different parts of China, reveal a lot of different tick-borne, tick-associated viruses. And we find more than a few hundreds of new tick-borne virus strains. And an interesting observation is that these tick viruses that we find, they actually, is located in a more kind of ancestral positions in the evolutionary history of the RNA viruses compared to other arthropod-borne viruses. And so that it means these tick virus, they have a more kind of important role in driving this early evolution of the viruses to infect the vertebrate host. And through this nationwide survey, we are able to characterize the different distributions of pathogenic tick-borne viruses and in different tick vectors. And you can see they actually the different tick virus, they have a different distribution and preference. And some, for example, Jingmen tick virus, they are preference in the distributed in across different tick species, whereas some others, they tends to be more specific to tick species. And some viruses, this is the list of kind of relatively recent identified novel tick viruses that infect human. For example, the Yezhou virus that initially reported in Japan 2014 also keep reporting in other places recently. For example, in China also report these Yezhou viruses. And so the tick actually the tick-borne disease they move across the geographic regions, and of course human movement is one thing, but another thing is the animal host that where these tick-borne virus these ticks may infestate, the animal will also bring the virus to move from place to place. And we also look into what kind of factors driving the virus compositions in ticks, because that will help us to basically assess the risk of the different tick species. And we find that some the virus compositions, they are significantly determined by the ticks genus, and some ticks genus, they have a higher virium diversity than the others. And we particularly look into a important regional important tick vector, the Asian longhorn tick species, and because it carry the SFTSV and also other important tick-borne diseases. And we find that and also because of there are much more studies of this compared to the others. And we find that the chimerism factor is a strong predictor of the virium of this tick vector. And then because using these models that we know, we estimate that the chimerism factor is an important driving of the ticks driver for the ticks virium diversity. We can use this information to predict the global trend of the tick virium diversity based on the understanding of predictions of how climate changes over time. And we find that more than 80% of regions will have increased tick-borne diversity by 2030. Of course, a lot of this data came from the training data came from the Asia regions, and so that there are a lot of knowledge gap of different tick-borne virus diversity and also tick diversity as well in different other regions. And so it's important to have this data in order to know better, to have better predictions. And so the vector itself is an important driver of the vector-borne diseases, and so our team are also working on looking at the vector genomics to understand how the vector actually interact with the pathogens, and then drive the emergence of the diseases. And for example, this one, we basically find that a lot of quite a sum of the mosquito genes in Anopheles actually is under selection pressure, and then kind of co-evolving with the parasite that they are, the pathogens, parasite pathogens they are carrying, and then these might open a new research target to understand which genes in the mosquito may actually facilitate the transmissions of these arbor pathogens. And the understanding of vector, genomic and also phenotype, particularly, for example, their susceptibility to insecticide, to pest control effort is also important. For example, insecticide is one of the very major approach to control mosquito in the world, and so in different part of the world, and but heavy use of this will lead to increase of the resistance. And we have done a survey in Hong Kong showing that in Hong Kong there are also populations which already develop resistance to certain insecticides. So understanding the vector can inform the better pest control strategies. And by also doing a lot of genomic sequencing, understanding the vector genomes is also important. What here I want to say is that this is one of collaborations effort with mainland Chinese team, sequencing quite a lot of different tick species. However, still now, there are so many tick species still without whole genome data. And although we do see some interesting geo-environmental situation of genotype that actually determine their virus carriage, but the lack of the genomic data in general actually cannot more such data would allow us to investigate this type of determinations of the virus preference in a deeper manner. And there is also one of the very big knowledge gap in tick-borne diseases. Compared to mosquito diseases, there are a lot of host factor, vector factor has been studied. But for tick-borne diseases, the tick itself and also the host factor like human genes that are involved in the tick-borne virus infections, there are a lot that is unknown because first, the tick itself is quite very diverse. There are many different tick species that can carry tick-borne viruses, and also also many tick-borne viruses as well. And so we are actually we have a team led by my teammate Dr. Luanda Lan, working on the understanding of host factor of mosquito pathogens, and we are basically transferring this type of platforms to try to understand the host factor for tick-borne viruses. And we managed to get a grant, and then a lot of such activity will be done in the next few years. So collectively, so this is a basic summary of my presentation. Collectively, the complexity of the vector-borne disease is due to the diverse vector and the pathogens involved. Of course, the climate and all the different factors we drive the emergence of the disease, and new pathogens and vector remain to be discovered. And one important knowledge gap is that many of the animal reservoirs are unknown, and the host and vector determinant and factors for infectivity, transmissibility of this vector-borne disease are really lacking. And so it requires basically, as you can see, this is not just human patients and then transmitting the pathogens among themselves, but it also involved animal, involved the affected organism as well. So it really truly requires an interdisciplinary collaboration between the clinicians, ecologists, modelers, virologists, bacteriologists, and so I recall that this conference, conference has a subtitle of "Collaborative Response to Fresh Challenges," and I think this is really, vector-borne disease really require a collective response, and we need this type of collaborations to generate understanding in order to provide a holistic approach to inform the vector-borne disease control and preventions. And so I would like to thank the Center for Immunology Infections, particular fundings from in Hong Kong, the Hong Kong governments to support the mosquito research that my team has been doing on surveillance, therapeutics, prevention, and diagnostics. And the work cannot be done without their fundings. And also the Jockey Cup Global Health Institute funding, and a lot of very great helps from our teammates and also collaborators overseas, and also the upcoming research funding that we have on tick-borne diseases. All right, so here's come to end my presentations.
Thank you, Professor Tommy Lam. Professor Tommy Lam is from the Division of Public Health Laboratory Sciences, University of Hong Kong. Is there any questions on site? None. There's one online, if you don't mind, stay a little bit longer, Tommy. The question is, just curious if Lyme disease, given lobster's tick presence, has been detected in Hong Kong, and given possible controversies in existence in Australia, diagnosis, management, and prevention, what are your thoughts?
Yeah, this is really big questions. So in general, Lyme disease in Hong Kong is not a particular very important very serious issue, although it has been found and the mostly, the control measure would include, for example, the tick surveillance. We know, understanding whether the disease vector is present, and also the diagnostic in the clinics with travel history, and so. So I think and the recent advance in diagnostic has been so good that a lot of these type of tick-borne disease can be picked up, and also vector-borne disease can be picked up. So I think it shouldn't be a big issues. But of course, there are some new kind of new pathogens from other ticks-borne viruses. They may require more kind of advanced diagnostics. But yeah, so in general, I think it's going on the right track.
Thank you, Professor. Okay, so our next presentation is going to be by Professor Sook San Wong, and join us to welcome Professor. Professor Wong is from the Hong Kong University's School of Public Health, and her presentation is going to be ISRV, Hong Kong School of Respiratory Viruses for Early Career Scientists.
Thank you, John. Good afternoon. I'm the last talk before lunch, so I'll try and keep this short. So today is not going to be heavy on the data. I'm an assistant professor at the Hong Kong U Pasteur Research Pole, but today I'm talking to you in my capacity as the member of the Education Committee of ISRV to introduce to you the ISRV School of Respiratory Viruses, and give you a bit of background, what we're trying to do, and then sort of do a bit of promotion, as well as let you know the initiative that we're doing to try to train the next generation of scientists. So some of you are very familiar with ISRV, and to those of you who are not, so ISRV is the International Society for Respiratory Viruses. It was actually started in 2005 as the International Society for Influenza and Other Respiratory Viruses, but post-COVID, we rebranded so that we can include other respiratory viruses into our activities as well. And so we rebranded as the International Society for Respiratory Viruses. Today, our society has more than 1,200 active members representing academics and healthcare institutions across many different countries. So the director is Maria Zambon, and she's also the director of the UK Public Health Lab. And that's the executive committee in that over there. So the global mission is, like APACI, is to relieve sickness and preservation of health by promoting prevention, detection, and treatment and control of global respiratory viruses. And we do the aims are we try to achieve our goals through these aims, which is to promote sharing of information and knowledge. And the second aim is to support earlier career scientists, particularly in the lower middle income country, to basically also address the global health and research disparity. So some of our activities, I will not go too much into it. Some of our activities is our flagship activity is to host the Options series of conference. I'm sure some of us have attended, and this is the biggest conference on influenza and other respiratory viruses that's held every 2 years. There are also other scientific meetings based on special interest groups, and we have the neglected influenza viruses special interest group, the epidemiology, the antiviral, and so we do have different specialty subgroups that also organize their own meetings. They are much smaller in scale compared to options, but we do have these activities as well. And then there are some grant, we call them grant-making activities, but they're basically efforts to try and increase our capacity to attract international funding. We have training events, and the school is one of these training events. And we also, we partner with Wiley to also host our journal, which is the Influenza and Other Respiratory Viruses. So this ISRV education program is officially started this year, actually last year, late last year, and it's led by our chair, Rebecca Cox, or Becky Cox, as some of you may know her, from the University of Bergen, Norway, and we have hired a full-time education manager. And we are supported, and this came about because we, Becky managed to successfully obtained a grant from Flu Lab to support 3 years of educational and outreach activities. And our core objective under this Flu Lab remit is to expand the school outreach to underrepresented region within 5 years to foster equitable access across diverse genders and backgrounds. So a little bit on the background of ISRV schools. The school aims to provide early career scientists with comprehensive understanding in epidemiology, molecular biology, immunology, and pathogenesis of respiratory viruses. We cover control and prevention strategies. We provide a forum for students to interact with senior scientists, and more importantly, with each other, so that we can sort of foster network opportunities. And also, ideally, we can facilitate the creation of global alumni network and continued education program. So there's two formats to the school. We have the full school, which is a 5-day residential program, as well as the mini schools. So the full school is an immersive, in-depth training program covering multiple different disciplines, and we usually provide accommodation, and it's delivered by partnering local institutions or research or universities. And as I've said, we cover multiple disciplines in our curriculum, and the activities will include lectures, workshops, presentations, mentorship, and networking. So essentially, we covered curriculum as well as soft skill development. For the mini schools, they are short focused programs that usually are linked to major scientific meetings like options, and it's designed to prepare early career researchers for conference participation, and we combine scientific contacts with professional skills and networking opportunity. So the next mini school will be tagged on to options meeting in DC, and it will be a 1-day program. So a little bit on the ISRV school. This is our 7th iteration, and it started in Siena by Emmanuel Montemoli, and it started as a summer school. And so over the years, we've been having them in different parts of the world. Hassan Zaraket organized it in Lebanon with the American University of Beirut. And then we had one in Vellore, India, with the Christian University, and then Christian Medical University. And then Qatar University, the University of Qatar with Hadi Yassin was the last one, late last year, I think, yes. And then in Hong Kong, which would be the 7th school, and it will be the first one that's going to be operated under the Flu Lab funding. So what we're doing with the Hong Kong school is that we're trying to be a bit more systematic and comprehensive by developing a program, an educational curriculum that we hope that the ISRV can put forward and bring on to the next other future schools. So we've had over 350 early career scientists that's been trained in different parts of the world since 2011. So as I've said, the 7th school in Hong Kong is the first to be held in East Asia, and the overall theme will be structured to focus on challenges within the region. However, one of our ideas is that we want to train future leaders with global perspective. So if we cannot send them to all the conferences in the world, then we're going to bring all the leading experts from different parts of the world to teach them and tell them about global challenges and experiences. But one of the unique reason why we wanted to hold it in Hong Kong is also to leverage the city's pivotal role in public health history in zoonotic outbreaks. And I think there's a lot to be learned from Hong Kong's experience in handling and especially control and prevention of infectious disease outbreaks. Ultimately, to sum up, our goal is to train, but also to inspire scientists with broad perspectives and creative solutions. So we're not thinking about, 3 years, we're thinking maybe 5, 10 years in the future, our cohort or our alumni is going to, by equipping them with good, broad knowledge early in their career, they are eventually going to come up with, good creative solutions to our challenge. So it's going to be a collaboration, and with primarily it will be hosted at the School of Public Health in Hong Kong U. But we are collaborating with Hong Kong U Pasteur, which is where I'm from, and Vijay as well. And then we're also partnering with GHI, the Global Health Institute, whose mission is also to address global health challenges. So in that sense, the missions are quite aligned between ourselves and GHI. So as I've said, the program core themes will cover multiple disciplines, which the faculty has consider to be essential in understanding how to control and prevent respiratory viruses. So you'll have virology, epidemiology, modeling, therapeutics and vaccines, and we'll cover zoonotic challenges as well. And then we also have a day that's focused on the policy and social sciences. This is how the draft program looks like right now. Each day is allocated to the themes that I've talked about. So the first day, students will be exposed to the overview and progress and challenges of, again, seasonal respiratory viruses. Well, actually seasonal, but we're actually going to include a zoonotic component into day one. Essentially, it will be a more global outlook on the landscape, the vaccine landscape, the antiviral landscape, the challenges across the different parts of the world. Second day, we'll go deep dive into the sciences. We'll talk about the virology, pathogenesis, immunology, and virus evolution. And then the third day, we'll talk about epidemiology, modeling, and public health impacts. And the fourth day will be all about zoonotic viruses as well, global health challenges. And we also have a field trip built into this day as well, where we'll take them to the Hong Kong Museum of Medical History, where they also have a special exhibit on the SARS outbreak, actually. Hopefully, we'll also try to build in a plague walking tour for our student. On the last day, we'll talk about social sciences, how to translate science into policy, as well as letting the students do the talking now by having some presentation. So some of the interactive activities we've planned would be 2 poster sessions, 2 workshops, one given by Vijay on phylogenetics, and the second would be vaccine strain selection. So we want to demystify how vaccine strains are selected, and this would be ideally, I think this will be run by Richard Webby. And then we will have the field trip as well as the student presentation and mentoring sessions. As I've said, we will have a mentoring session. We also have some career talks as well as grant and scientific writing. So we're hoping to build that in during the lunchtime. So ideally, we want the students to develop critical thinking ability and networking skills. These are some of our faculty, Joe, and some of them are also speakers from here today. And then just a brief overview on our school's learning objective. So number one, we want them to integrate foundational knowledge across all the disciplines. The second, we want them to apply analytical and problem-solving skills to interpret data. We give them the resources to interpret these data. And then we want them to develop effective communication skills. And then we want them to also establish future collaboration. And we are trying to do this in a regional basis. So most of our participants will be targeted for ECRs or mid-level career scientists within the region. I talked a lot about it, but unfortunately the registration is closed. So, sorry. I do want to tell, one of the goals is also basically to tell you how attractive this program is going to be so that you will be interested in our future programs and activities. So for our school, we've had a tiered registration. So as I've mentioned before, our funding is from Flu Lab, and which one of the goals is to support scientists from LMIC. So we have a tiered registration. So for high-income country, it will be 3,500 Hong Kong dollars, and the low-middle-income country would be 1,000 Hong Kong dollars, and this would include 5 days of residential accommodation, course material, most daily meals and local transport, as well as ISRV membership. So it's really high value, for what you're paying for. So far we've had 88 participants from 27 countries with 33% LMIC participation, and we've dispersed 42 scholarships. So sorry, registration's kind of closed, but these are our upcoming schools. We will have a residential school in Africa. We are working on that now. The date is not confirmed yet. And then in 2028, we'll have one in Chile. And the mini schools, the upcoming mini schools next year is in RSV, after an RSV meeting in Spain, as well as the second mini school would be after the neglected influenza meeting, which will be planned in Cambodia. So if you're interested, do scan this QR code to register your early interest. And so some of the participants who've emailed us after the closing deadline, we've put them on our waiting list. So if you're interested, do scan this QR code, put yourself on the waiting list, and we will give you priority to register for the next meeting. So yeah, that's my concluding slide. So I'll just leave you with our mission statement, which is to develop the next generation of global respiratory virus experts, and that's the QR code if you want to email us for future for further information. But I would like to also put it out there to the members of today's meeting. We've got funding from Flu Lab for the next 3 years, but if you think this is good value, then perhaps that something could be done with the members of APACI on how to carry on this program beyond the 3 years for this region. So ISRV has established this template. We have the alumni, we have the know-hows, we have some of the logistics. So if you're interested in sort of hosting similar programs, potentially this could be something where we could partner up and sort of think about how we can benefit, the future scientists in the region. Just a few slides for the upcoming ISRV activity. So we have options in DC. I think the late registration is still open. And then there's the RSV meeting in Spain next year, as well as the planned meeting school in Chile in 2028. I do have to say that because of the funding sort of structure, the schools tend to be focused on the region. So if it's going to be in Chile, I'm not sure participants from the Asia-Pacific region would be prioritized. But if you're still interested, please do contact us, and then maybe we can work out something. Thank you.
Thank you, Professor Wong. Is there any questions, comments, applications?
I should say it's not only meant for early career scientists, it's meant for anyone who wants to start in respiratory virus. There could be mid-career scientists who decided that, I want to go into respiratory virus. So this might be a good program or platform for you to sort of get your feet in the door, get to know the people who are working in this field, and also for senior scientists who just wants to a refresher training to be up to date with what's happening.
It'd be great to see that there is actually collaboration between ISRV and APACI. You want to say something about this?
Yeah, thank you so much. And thank you for taking up the invitation to come and speak to us about this today. I think the fact that the school is happening in Hong Kong is great. I think this feeds into a broader involvement by ISRV in the region over coming years. And I'm definitely, on behalf of APACI, can say we are interested in finding a way for this to functionally move forward in a way that can keep our young researchers engaged and have respiratory diseases, respiratory pathogens be something visibly attractive to pull in some of our best talent and keep the pipeline full.
Yes, John, thank you for the invitation to speak today as well and to meet members of the APACI. And I do share your sentiment. I think we should work together to sort of like, for the benefit of the region, right? Yeah.
Thank you. So now we're open to have lunch. Thank you very much for being here. And lunch will be served on the outside where the coffee was, and we have to return by 1:45. Okay. Thank you.